A-Level Biology 9700, Paper 4, Variant 2, February March 2025. Question 3. Gentamicin is an antibiotic used to treat severe bacterial infections in children. Some children have a genetic mutation in the gene MTRNR1. If gentamicin is given to children with this genetic mutation, it can cause deafness. So before gentamicin uh, can be given to a child with a severe bacterial infection, PCR, polymerase chain reactions, and lactophoresis are used to test whether the child has this mutation. If the mutation is found, different antibiotics must be given. So describe and explain the role of Taq polymerase in PCR. So very simple question is there, that is directly they have asked the uh, role of Taq polymerase in PCR polymerase chain reactions. So the role of Taq polymerase in PCR in polymerase chain reaction uh, is that is the Taq polymerase is a crucial enzyme responsible for synthesizing new DNA strands. So I would like to write on the side. Polymerase is an enzyme to synthesize new DNA strands. So, Taq polymerase enzyme uh, is responsible for making or for synthesizing new DNA strands. So, these different steps, which can be, you can say, considered for its role. First of all, the first is the denaturation. For polymerase chain reaction, uh, the denaturation is the PCR process, uh, which begins with heating the DNA sample to a high temperature. The high temperature heating the DNA sample to high temperature. And you know, at high temperature, the DNA will be denatured. And that temperature can be 94 to 98 degrees centigrade. That is too high temperature. So this high temperature will breaks the hydrogen bonds. It will breaks it will break hydrogen bonds, and you know, the double strand DNA will separate into single strands, and it will leads. I'm just putting the arrows, and you should also listen my words which are I'm explaining with this writing. So denaturation will occur when there will be the heating up to 94 to 98 degrees centigrade. So the hydrogen bonds will break between DNA strands, and the DNA strands will separate into single strands. And then after this, the second step is annihilation. So annealing is the temperature is lowered. Temperature is lowered, and the temperature is lowered 50 to 65 degrees centigrade. So it will allow the short DNA sequences primers to bind the complementary regions on the single strand DNA. So what will happen during this? That is the short DNA sequence or primers will bind on complementary regions on the single strands. Then next is extension. Next step is extension. And during extension, the tag polymerase will come into play. So the temperature is raised to the optimal activity again. The temperature will be raised to high temperature, you can say, to its optimal activity, raised to 72 degrees centigrade. So tag polymerase then binds to the primer. So it will bind to the primer. Base pairing will occur. That is, the adenine will join with the thymine and guanine with cytosine to synthesize new DNA strands. So then there will be the repetition of all these steps, that is the denaturation, annealing, and extension. So in this way, you can see the tag polymerase is used in PCR is its heat stability. Tag raise is used due to its heat stability. That is, the high temperature is required during the PCR reactions. So the tag polymerase can bear the high temperature. So high temperature, when it is paired, so it will be helpful to perform this whole process. So it has adapted for this high temperature. So this adaptation allow tag polymerase to withstand the repeated heating and cooling cycles of PCR without denaturation, stability, and will not denature during PCR reactions. PCR repeating reaction. So these reactions are repeated again and again. So it will withstand with the high temperature and with the lower temperature as well. So the next question is, PCR with primer specific to the MTRNR1 gene is used to amplify DNA from the child that is being tested. The PCR primers are designed to so that the amplified product of the normal allele of MTRNR1 is longer than the amplified product of the mutant allele. Gel electrophoresis is used to separate the PCR products. So figure 3.1 shows the results of gel electrophoresis after using this method of PCR on DNA samples collected from 3 children. So they have given us the child 1, child 2, and child 3. So direction of DNA movement, and question is state which of the children in figure 3.1 cannot receive the antibiotic gentamicin to treat a severe bacterial infection. That is, only the gentamicin is given to those which are suffering with this infection. So gentamicin, antibiotic which can only be used for the children who are suffering with infection. So here in this case, you can see they have shown us the 3 child, 1 child 2, and child 3. So these they have given us. So here I would like to say that we have to only the child which will not have this infection and will not receive any antibiotic gentamicin. So here you can see child 1. The child 1 uh, has only single band. You can say the single band, which is at higher level. You can see which is at the top. So it indicates the presence of indicating the presence of only the longer DNA fragment. And it means it has 2 copies. This child has of normal allele of the MTRNR1. So this child uh, will have normal. And the on the other hand, you can see show a single band at the bottom. This child 2, single band at the bottom. So it indicating the presence of only the shorter DNA fragment. So shorter DNA fragment and shorter DNA fragment means mutant, it corresponds to the mutant allele that suggests that child 2 has 2 copies of the mutant allele. It has 2 copies of mutant allele. This child has genetic mutation. And this child 3 has both, shows 2 bands. One at the top Top shows the normal, longer DNA or the allele. You can see that. And the A. And lower one shows the mutant copy of allele. And we can say that it will act as a carrier. It is the carrier. He is the carrier. Carrier of genetic mutation. So these are the explanation of these bands which they have shown. Now we are moving to the answer of the question in the light of these descriptions which I have given to you. That is the state which of the cannot receive the antibiotic to treat a severe bacterial infection. So you can see it will be the child 2 because it has the mutant alleles. Next question. How gel electrophoresis produces the pattern of results shown in figure 3.1 from the PCR products of the MTRNR1 gene. So we are moving that answer of this question. Well, so we have to break down the how gel electrophoresis creates the distinct banding pattern, which is observed in figure 3.1. So the process of electrophoresis, gel electrophoresis and DNA separation. Electrophoresis separates DNA fragments based on their size and electrical charges as they may move through a gel matrix under the influence of an electric field. So I would like to write over here that is the gel separates DNA fragments based on their size and electrical charge. As they move through gel matrix. And the electric field is applied over there. So here the the child one single band at the top of the gel indicates that all the amplified DNA fragments from child one are of the same larger size. Since we know that longer of fragment corresponds to the normal allele, this means child one only has the normal allele of the MTR and R1 gene. So these longer fragments experienced more resistance in the gel and thus migrated shorter distance, and the child 2, you know, uh, the single band at the bottom of this indicates that all the amplified DNA fragments from child 2 are of the same smaller size. So the smaller size corresponds to the mutant alleles, meaning child 2 has only the mutant allele. So these smaller fragments encountered less resistance and migrated a greater distance through the gel. So child 3, upper or lower, the presence of the two distinct bands, one top and one at the bottom, shows that child 3 has two different sizes of amplified DNA fragments. The upper band represents the longer fragment from the normal allele, and the lower band represents the shorter fragment from the mutant allele. So these two populations of DNA fragments separated based on their size as they moved through the gel, resulting in two distinct bands. So in essence, gel acts as a molecular sieve. Um, they will it will I am going to write, first of all, I would like to, that is the child for the child 1, answer for the child 1. They had normal allele due to which experienced more resistance and covered less distance, and child 2 had mutant allele, so faced so faced less resistance and covered more distance. And child 3 faced both and showed two bands. So all this we can say that this process uh, which is called as gel electrophoresis acts as a molecular sieve. So it filters, or you can say the filter, separating the PCR products of the mTRNR1 gene based on the size difference between the normal and mutant alleles. So it showed the uh, different distance and size, so thus producing the distinct bending pattern that allow us to determine each child's genotype. For this specific, so it is very helpful in filtration of the different bands of the DNA alleles. Now, next question is there. Some bacteria have plasmids that contain a gene conferring resistance to gentamicin. The gene can be transferred to other bacteria. Suggest how the gentamicin resistance genes can be transferred to the other bacteria. Well, there are certain methods you can say by which the gentamicin resistant genes can be transferred uh, from one to the other. So for this purpose, you can say the first process which can be it is conjugation, which occurs in bacteria. So during conjugation, the is a direct transfer of genetic material between bacterial cells. When physically in contact. As for example, you can see they are very, and then the genetic material is transferred from one to the other. So cell to cell contact, on the other hand, we can write the cell to cell contact. And then third, which I want to write is plasmid transfer. Cell to cell contact, they will the cells will come close to each other and they will exchange their material. So the plasmid carrying the gentamicin resistance gene is replicated. So in this way, it can be transferred. So these are the 2-3 different ways by which. So it was all about question number uh, 3. Please like, share and subscribe my channel. Thanks for watching.